Professor Sudarshan sir, Dr. Ravi sir. On behalf of the Vice Chancellor, Registrar and other statutory officers of the university, I welcome all of you to this training program on COVID-19 from an initiative from Rajiv Gandhi University of Health Sciences. I hand over the floor to Professor M.K. Sudarshan, Chairman, Technical Advisory Committee, Government of Karnataka, for his opening remarks and introduction of the speaker. Over to you, Professor Sudarshan, sir.
Thank you very much, Dr. B.J. Mahendra. Thank you, Dr. Dayanand and
Yes, thank you, sir.
Dr. Rupashri. So I am extremely happy to introduce my colleague, Dr. V. Ravi, who is popularly known as Virus Ravi. He is not offended by this statement. In fact, he is very proud to say that he is Virus Ravi. I have known him for nearly 3 decades since his career in NIMHANS, and he is presently the state nodal officer for genomic sequencing of COVID-19 virus. He has worked in the field of virology associated with SARS, H1N1, Ebola, and many other viruses. And he is also a member of the State Technical Advisory Committee, my colleague. And also he is very interestingly and importantly, we must know that he is a member of the research committee on the International Committee for Sputnik vaccine, Russia. Okay. And he is also in many national bodies. And today his lecture is very apt because for all practical purposes, as this is a scientific assembly, I can say that we are swept away by the second wave of COVID-19. And one of the reasons that is largely attributed is so many variants which are considered very infectious and some virulent. In fact, I am also very curious to listen to his lecture. So with this short introduction, without any further ado, I request Dr. V. Ravi to deliver the lecture. Over to Dr. V. Ravi, please.
Good afternoon, sir. Thank you, Professor Sudarshan, for your kind words. Let me thank Rajiv Gandhi University of Health Sciences, Honorable Vice Chancellor Dr. Sachidanand for giving this opportunity. Also want to thank Dr. B.J. Mahendra, Dr. Dayanand, Dr. Gautam, and Dr. Rupashri for facilitating this program. What I will do is in the next half an hour, try and tell you what little I know about SARS-CoV variants, share the facts, and then probably try to bust some myths that are existent amongst medical fraternity as well as general population. The outline of my presentation is I'll give a very short introduction of SARS-CoV-2 structure, genome organization, classification and nomenclature that will lay the foundation for the lecture. Then I'll answer questions on what are variants, why do they emerge, how do we detect them, what is the current status of variants globally and in India, what are the implications of these variants, something which Professor Sudarshan very nicely alluded to in his introductory remarks, a few myths related to variants, and end with a summary. We all know coronaviruses have been around for 50-60 years. They're a family of RNA viruses that are enveloped, distributed widely between humans, mammals, and birds. 4 coronaviruses, 229E, OC43, NL63, and the HKU1 have been known for almost 5 decades. They cause common cold-like symptoms. the major severe acute respiratory coronaviruses emerged in the turn of the century. The first one was called SARS. It again emerged from southern China and spread to almost all continents. Fortunately, India was spared. It was contained well with many containment measures. In 2012, a related virus emerged from Middle East known as Middle Eastern Respiratory Coronavirus, which causes Middle Eastern Respiratory Syndrome. This is still circulating, but largely restricted to the Middle East. Late 2019, a new coronavirus emerged, and on January 5th of last year, the virus was completely sequenced. Very interestingly, this virus was detected by whole genome sequencing first, rather than classical virology of isolating the virus and then trying to characterize it. Directly from clinical specimens of people who are coming down with a severe acute respiratory syndrome, specimens were subjected to next generation sequencing, and they identified the sequence similar to SARS, but it had several changes. They're widely distributed in nature. Their genetic diversity, they're called corona because they look like a crown under the electron microscope, studded with its spike protein on the surface. Coronaviruses are known to spill over from one species to another. Natural reservoirs are bats and mice. The SARS-CoV-1, the one that emerged early this century, was a spillover from bat to civet cat and then to human beings. The MERS coronavirus was a coronavirus that spilled over from bats to camels and then to human beings. SARS-CoV-2 is a spillover from bats to pangolin, an anteater-like animal, and then from there to human beings. And these were sold, both bats and pangolins were sold in the wet market of Wuhan. So spillover is known. Recombination also is known in coronaviruses. It's a fairly large RNA virus, measuring 60 to 120 nanometers. It's spherical. Its surface is studded with a very important protein that has a globular head and a linear stem. On the left, it is represented in the cartoon as a yellow protein with a like a common pin. On the right, you can see it as a red protein jutting out of the surface of the virus. Now, this is the most important protein because almost all properties, biological properties, are associated with this virus. And this protein has a globular head and a stem, and in the head, a region here that has been enlarged is one particular portion known as receptor binding domain, RBD. Now, receptor binding domain is unique to SARS-CoV-2. It's different from SARS-CoV-1. Remember this because the neutralizing antibodies, the antibodies that neutralize virus infection, are directed to the receptor binding domain. And very interestingly, many of the new variants that are emerging have mutations in the receptor binding domain, in the amino acids in this particular domain. The genome is approximately 30,000 nucleotides long. It's a positive stranded RNA. It codes for several genes, two major open reading frames, ORF1A and B from the 5' end. Then comes the spike protein gene, and then you have the envelope, the matrix, and the non-structural proteins are interspersed in the 3' half of the virus genome. You can see in the bar below that the receptor binding domain is somewhat in the center of this whole genome, and that is where the nucleotides coding for the spike protein gene are. Okay, generally, most of you know, based on genetic sequences, people do comparisons, and when they comparisons, they use software to see relatedness. This is a dendrogram. And those of you who are interested in your family history and anthropology would also like to make dendrograms. So and so were husband and wife. They were the origins of the family. Children were so and so, and the tree goes on. Here also it's a tree like that. The novel coronavirus 2019, you can see in this tree, sits in the center. The closest relative is the bat coronavirus. You can see way up is the SARS-CoV-1. So it is distant from the SARS-CoV-1. So this relatedness is very important to ascertain relationships between various virus sequences, and that gives you a picture on how the whole genome is evolving because of mutations. Okay. There is a lot of confusion in people about the way names are given to lineages. The most common ones circulating in WhatsApp University uses the name of a country. That is not the correct scientific way of calling a lineage. It is said UK variant, South African variant, Maharashtra variant, Brazilian variant, as if that country has contributed to the emergence of the mutant. That is not correct. Those are the countries which first detected the mutations in the virus. Nobody knows where these mutations originated because we are not doing sequencing continuously on a regular scale across the globe. There are many nomenclatures used for lineages. All the sequences of SARS coronavirus 2 are hosted in a global database, which is called GISAID. You can see in red letters in the second paragraph of this particular slide. GISAID stands for Global Initiative on Sharing All Influenza Data. This was started in 2006 to collect data of influenza virus sequences, including avian influenza virus sequences. Slowly, the database became so big, so all the other new viruses also started getting added onto this big database. Now, SARS-CoV databases, virus sequences are also put in this database. There are various classifications. There is a GISAID classification, which gives certain alphabets to the lineages. But what people are using overall is a lineage, the nomenclature derived from a particular software known as pangolin software. Now that is given, that is used extensively by people. And in Nature, sometime last year, there was a paper published saying that the pangolin lineage is versatile and you can go on adding newer variants, and it is so versatile, so people started using it. So most of the people now use pangolin lineage. Now what is lineage? Let me explain to you. So this is a lineage that uses alphabets, and as time evolves based on the mutations in the virus, the tree goes on expanding. There are three major groups in the pangolin lineage: A, B, and C. A refers to the parent Wuhan virus, which was isolated from China. Then it went to Europe and America, and there it started mutating. And when the mutations were beyond a particular frequency and percentage, a new lineage called B was formed. B started mutating, and there are certain generic characteristics in B. And when they started changing, they called it B.1. And B.1 started mutating further, then they started calling it B.1.1, and so on and so forth. So B.1, then B.1.1, 2, 3, 4, 5, 6, and so on. Ultimately, there's another large group that has emerged now, that is the C virus. So this is the way the hierarchical system is used for giving the lineage identity to viruses. Now, this is a map that shows you the parent Wuhan virus in a color-coded form on the top. And as of now, in April end, from the original Wuhan virus, there have been 4,400 amino acid substitutions found in coronavirus genomes. That's a lot. That's a lot of changes in amino acids. And at the bottom, you can see a line diagram, a Manhattan, which tells you where are all these amino acid changes. The red portion here is the spike protein. Most mutations are occurring in the spike protein. That is something I wanted to highlight to you. Why do viruses mutate? Okay, viruses, how do they mutate? Viruses generally acquire mutations over time. And when these mutations occur, they are called emerging variants. These variants that emerge are important for a variety of reasons. As Dr. Sudarshan said, A, increased transmissibility, B, the variants as they emerge, they can escape neutralizing antibodies because their conformation is different, their shape is different of this protein. They evade natural immunity also. And because of evasion of immunity, vaccinated individuals can get infected. We are seeing, many of us know somebody who has had vaccination, and there is a fear that diagnostic tests may miss. Fortunately for us, most of the tests that we use now are not dependent on spike protein gene in RT-PCR. So this is still not a problem for us, but it can become a problem if other genes start mutating. If there are antivirals, they can escape activity because of the mutation. And mutations are also associated with change in disease clinical phenotype. That is a worry. We know that there is a very severe condition in children known as multi-system inflammatory syndrome following corona, MISC. Now that is seen in a small proportion of children. It's a life-threatening condition like Kawasaki syndrome. So when mutations are associated with that, and also mutations associated, may be associated with higher mortality and morbidity. So any mutation that is appearing is referred to, these mutations are referred to as variants. Now there are two types of variants that people talk about: variants under investigation or variants of interest, and variant of concern. Let me explain what the difference is. A variant of concern is a variant that has extensively spread across an entire nation or across nations, and there is enough epidemiological evidence and or cell culture evidence to say that its infectiousness is more than the parent virus. So you need cell culture evidence. You also need epidemiological evidence that this variant is infecting more people compared to the parent virus, what we refer to as R0. So R0 should be higher for a variant to be called variant of concern. If these evidences are not there, but we see it is widely spreading, then it is classified as suspicious, either as a variant under investigation or a variant of interest. So I hope I made things clear to you. Why do variants emerge? Mutation is a gift given by God to RNA viruses. All RNA viruses mutate because they have an enzyme known as RNA-dependent RNA polymerase that actually copies the genetic information in replication. And when it does that, it has poor proofreading ability. So it makes mistakes. So when a strand replicates of 30,000 nucleotides, you can have one or two nucleotides wrong strain. As more and more multiplication occur, more and more errors are introduced into the new nucleic acid that is synthesized. So this is an inherent advantage for virus that it mutates. The second reason why viruses mutate are because of immunological pressure. When viruses infect an individual who's been exposed or who is being treated with passive immunotherapy, the antibodies put pressure on the virus and the spike protein mutates, and therefore new variants are selected based on the Darwinian principle of selection of the fittest. So that is the two reasons why viruses mutate. Now let me tell you, a mutation by definition is a change in nucleic acid. A change in nucleotide. A change in nucleotide may or may not translate into a change in amino acid on the protein, and all amino acids that are changed may or may not alter virus behavior. This is fundamental biochemistry. Go back and check your biochemistry if you have any doubts. If the amino acid that has been replaced is of a different charge and significantly different, then the biology and the behavior of the virus will change. So most mutations do not have an impact, but certain mutations in key areas will certainly have an impact in the way the virus behaves. And when we say behaves, we are talking of better attachment, better penetration. In virology language, it is called better fitness. So viruses in the last 1 year, SARS-CoV-2 viruses have evolved with better fitness to engage much more firmly with the receptor called ACE2 on the surfaces. There is a hypothesis of why the UK variant emerged. There is a history of a patient who was an immunosuppressed individual, and he was being given plasma therapy and he got infected, and because of prolonged passive immunotherapy and being an immunocompetent individual, the virus mutated and became the UK variant is one hypothesis. Moving on to how do you detect variants? Okay, variants are detected using clinical samples that come for RT-PCR, nasopharyngeal and throat swabs that come in virus transport medium are used before doing any method for detecting a variant, and the method is whole genome sequencing. And whole genome sequencing, before you do it, because it's very costly, because it's very time consuming, because it is very skilled, because it requires specialized equipment, one would again like to confirm the PCR result in the sequencing lab. That's number one. So if you refer a lab to a sequencing facility, they don't do it, it's probably because PCR repeated there is negative. Either sample is deteriorated or it is a low positive sample. The second condition to be fulfilled for sequencing is the sample should have a CT value less than 25, because this sequencing results are best when viral copies are more. Then only you'll get full length sequence. You will also get sequences of genomes for samples that are between 25 and 30 CT, but you will not get full length sequence. Best results are with samples less than 25. So it's the second conditions. And you need a large volume of the sample to be sent. India has set up a consortium of genomic sequencing labs. It is called Indian SARS-CoV-2 Genomics Consortium, or INSACOG. And this consortium has been set up with three objectives. One, to see if there are new variants that are in the country. Second, to see if these variants have any impact on the public health, namely spread. And thirdly, to see if any of these variants are associated with a huge surge or a super spreader event in a given area. The technique of sequencing that is being adopted in INSACOG is represented as a cartoon on the right. Currently, the INSACOG recommends testing samples for sequencing from all international arrivals because they come from other countries. So international travels who are RT-PCR positive, CT value less than 20, their samples are sent for sequencing. And this RNA is extracted, it is converted into cDNA, and then using a set of primers and barcodes, large number of PCRs are conducted. These are small bits of PCRs that actually span the entire 30,000. You can't do a one PCR for 30,000 nucleotides. So many small pieces, and then it is joined together, and then the machine starts reading the nucleotides and does it over and over, generates a consensus sequence, which is then using bioinformatic tools compared with other genomes in a database. And ultimately, the software generates a report whether it is a known variant or it is a new variant. This whole process will require approximately 2 weeks. You can probably finish it in 1 week, but the actual technique takes maybe 24, maximum 48 hours, but the bioinformatic processing and the data processing takes approximately 4 to 5 days. It's a very laborious exercise. Okay, these are the 10 INSACOG labs. What is relevant, I have boxed for Karnataka. You have the NCBS, INSTEM NCBS in GKVK campus in Bangalore, and NIMHANS Neurovirology Department. These are the two laboratories doing sequencing. The government of Karnataka, in a circular on the left, has identified 10 sentinel sites for sequencing. These comprise of 5 hospitals and 5 SARS-CoV-2 labs. This is based on the WHO guidance. When you do sentinel surveillance, you should do from samples from hospitals and from labs. The 5 hospitals are Victoria, Bowring, Wenlock, Belagavi Institute Medical Sciences Hospital, and Bidar Institute. The labs are NIMHANS Virology Department Lab, Mysore Medical College VRDL, Hassan Institute of Medical Sciences, Bangalore Medical College and Research Institute VRDL, and Gulbarga Institute of Medical Sciences. As you can see, the state has done a very nice job of having geographic representation of labs and hospitals spread across the population. Let us move on to the implications of variants. There are 3 major implications: 1 on its transmission, 2 on its replication, and 3 on vaccine efficacy. Now, this is the current global status. A is the parent Wuhan virus that emerged in late 2019, and then it went to Europe, it mutated there. One of the major mutations I will show in the next slide occurred early last year, end of February, and that was the B.1 first generation variant that appeared from the parent Wuhan virus. Now, this mutated further into several lineages. You can see B.1 is the parent. From B.1, you have several numbers emerging, and they have emerged in many, are detected in many states: United States, South Africa, California, New York, and then of course you have Britain, Denmark, the newer variants, and so on. So currently there are many lineages circulating globally. Whenever a lineage emerges, and if it is a variant of interest or concern, it will have better fitness to receptors, so it will start replacing the existing virus, and because of international travel, it gets dispersed all over the world. That is the cartoon telling you that story on the top. In the bottom is an example of B.1.7 or the UK variant, which was first detected in December as a problem in several clusters in Wales and southeastern England. It started spreading fast, and you can see in the time scale, it has moved across the entire globe, and it's continuing to replace existing local strains. The dark color here means large number of cases. India was pale green in January. Now you can see it is moderate green in this country. So these variants slowly take over. I was telling you that one of the first major mutations that altered virus behavior was this D614G. What is D? As you know, amino acids are represented with a letter. D is aspartic acid and G is glycine. At position amino acid 614 on the spike protein, the aspartic acid was replaced by glycine. That one amino acid change gave more advantage to the virus for better infectivity, better engagement with the receptor. And you can see from February middle to almost May 1st, it completely replaced the existing circulating virus in the world, and D614G took over. Now, this is the grandmother now for the current variants. Based on this D614G, several other mutations started coming. Incidentally, D614G belonged to lineage B.1. And one of the biggest variants of concern that emerged in November, December was the UK variant, which is called B.1.1.7. Now, currently, if you look globally, there are many variants that are spreading widely. 3 of them are called variants of concern. The UK variant, 1.1.17, the South African variant, B.1.351, the Brazilian variant, which is called P.1. Of course, the Californian variant, which is B.1.427, which caused a problem late last year in California and early this year. Now, on that Californian variant, a second mutation occurred, and that was detected in Maharashtra, which we fondly refer to as the double mutant, and internationally, it is stigmatized as the Indian variant. Now, these are the 3 ones of concern. They are of concern because there is evidence to say that A, their R naught is much higher. They are 40 to 60% more infectious. There is sound epidemiological data, and there is also cell culture data. But what we are concerned in India is the B.1.617 mutant, which is called the double mutant, which is also called variably as the Maharashtra mutant. This mutant currently has been identified in the last 4 weeks to 8 weeks in 36 countries. I'm giving you the top 15 here. Highest number of percentage sequences are detected in India, followed by Australia, and followed by Bahrain, and then Australia and New Zealand. No wonder Australia and New Zealand closed its borders for travelers from India. So it is now spreading globally. That's the point number 1. Point number 2, in the country, it initially spread a large amount in Maharashtra in various districts, but now it has been detected in several states, as many as 16 states as of today. This is 15th April data. You can see it has been detected in almost many northern states, and in south it has been detected in Karnataka. This is the way the mutant, this particular mutant, has taken over in Maharashtra. This is some bar diagram. Each color here represents a variant. You can see the color code given in the key on the right on the top. The maroon one is the double mutant. You can see in the last few months between December and now, the double mutant is accounting for almost 50% in Maharashtra, and so also in 50% in Delhi, You can see that black triangle going up, that is B.1 point. And this is a comparative graph from Bengal that shows you this red waves that you are seeing is the representation of the double mutant. And now in the last 2-3 weeks, the Maharashtra mutant B.1.617 and B.1.618, which is referred to wrongly as triple mutant. I'll come to that shortly. That is also a very close relative of the Maharashtra variant. Is taken over completely in Bengal. What about South India? This is from the INSACOG data. Steadily, you can see week-wise the numbers of the B.1.617. The Maharashtra mutant is increasing, and today it forms more than 50% of sequences in South. Now, about this myth about triple mutation. Now, this triple mutation is a misnomer, because in a analysis of data from GSAID database, it was found when people initially deposited these sequences, there were 2 other small mutations which were not in the critical region, and they were V382L or W152L. They were also there. Somebody found, oh, this mutation is there, and said this is a triple mutant. It has been there all along. So the triple mutant and the double mutant belong to the same lineage. I want to make that point very clear. We have been doing sequencing at NIMHANS. This was sequencing which we published during the lockdown period. At that time, we had found all these lineages, B.1, B.4, B.18, and we had one small lineage known as B.1.36, which was only 2%. When we started sequencing samples in December in a large scale, we found B.1.36 to take over. It became nearly 50%. But that did not last long. January, February, B.1.36 dominated. But since March beginning, B.1.67 has taken over. This is the current results of the NIMHANS and NCBS sequencing labs. Total 554 samples have been sequenced, completed from Karnataka, and 314 were neither UK variant nor South African nor the double mutant. So they were other lineages. The double mutant is taking over. I will show you a graph in the next slide. It constitutes approximately 1/4 now. And UK variant initially was restricted to travelers. Now we have evidence to say it has spread to the community. We have very few South African variants, and they are not causing much problem. Apart from this, there are samples pending that need to be completed. This will be completed probably in the next couple of weeks, because money has now been released for this project. Now, here is an evolution graph. I was telling you about B.1.36.29, a lineage that started somewhere in 2020. Slowly, end of the year and beginning of this year, you can see the proportion. It went up to 30% of all samples that we sequenced, and suddenly entered the Maharashtra mutant. Maharashtra mutant has now taken over, and I think the latest results are that it is almost 70-75% of all sequences that we are getting in samples in Karnataka. The other implications of variants, I would allow this is country-wise. What are the variants? So just to give you a flavor that almost many countries have variants, I would draw your attention to the right half of the table. There are 3 headings known as transmissibility, virulence, and antigenicity. We are talking about implications. Mutations and change in amino acids have implications for transmissibility. I gave you elegant examples globally as well as within our country. Now, what about virulence? Is there enough evidence to say the mutations and the new variants do they have any effect on the behavior of the virus in terms of disease causing potential? Unfortunately, majority of these variants, there is no evidence in change of virulence, and some of them are under investigation. This one, especially the UK variant, it's still under investigation, and so also one in the Brazilian variant in Japan. There are some studies going on. On the other hand, there are many studies that are actually available showing that to varying degrees, these viruses escape vaccine antibodies. Before I go to that, I was telling you in the beginning, to prove a variant is a variant of concern, one is field data showing that it infects more number of people and R naughts are more. The other is to compare replication in cell culture. Now, these are graphs that are showing you on the x-axis, the y-axis, the percentage of viral titer or binding, and on the x-axis is the amount of virus that you are actually detecting. And you can see the black solid line is the parent Wuhan virus. If there is a shift to the left, that means these are viruses that are replicating much more efficiently. They have better binding and they have better replication. The last panel is a signature mutation of the UK variant. You see the curve have shifted to the left. Now, L454R is one of the mutations in the Maharashtra variant. There are some papers. This is the Californian one, where the origin where the Maharashtra mutation originated, the backbone, and there also it is more efficient because there is a shift to the left. So this is one of the evidences that is required. Now, here is a paper published in New England Journal of Medicine very recently, which talks to you about the effect of variants and vaccination. Now, the rows are the different vaccines: Johnson & Johnson vaccine, Pfizer, Moderna, the Sputnik vaccine, the AstraZeneca vaccine, and then the Novavax, which we don't have in India, the Sinovac we don't have, and of course the Sinopharm, which we don't have. The summary and substance of this complex table is boxed for you for convenience by me on the right. You can say that the efficacy of some of these vaccines against variants is decreased. They do still neutralize variants, but the efficiency which they do is less. For example, Johnson & Johnson vaccine, UK variant, 57 to 85% in two different studies is the efficacy. The AstraZeneca is 22%. And the South African variant data is also available. I'm not showing you that. The South African variant is much less effective as far as AstraZeneca vaccine is concerned. But mind you, it is not that the vaccines are not at all effective. The degree with which they protect may be less depending on the vaccine, but they still protect against serious disease and death. That is something you should remember. What about our own Atmanirbhar vaccine, which is the Covaxin? The National Institute of Virology has done several studies, both in vitro and using serum samples of vaccinees from Covaxin. And it has shown that a person vaccinated with Covaxin, his serum, or a person who is infected and recovered from natural COVID infection, their serum samples, both are able to neutralize, you can see in the graphs below, to equal efficiency the D614G, the grandmother virus, or the Maharashtra mutant, or the UK mutant. And I think recently they have also shown for the South African variant and the Brazilian variant. So the news seems to be good as far as Covaxin is concerned, because it seems to neutralize, at least in vitro in the laboratory, equally. Moving on to the myths. New strains have made the virus more deadly. There are two kinds of myths. There are two pundits in WhatsApp, great professors. One group says, as virus mutates, it's become less virulent. Absolute armchair theory. There is no evidence to show. I showed you a table with respect to virulence. There is no evidence to say the virus is less virulent. Let me give a common sense point. People are getting infected with the Maharashtra mutant. Do they have less severity of disease? Is the pattern different? That only asymptomatics are coming with the Maharashtra mutant? The answer is no. So it is not becoming less virulent. Is it becoming more virulent? If it is becoming more virulent, we should have had large number of people getting admission into ICUs. The proportion remains 80 at home, 10 moderate requiring oxygenation, or 10 or 15, and only 5% are going to ICU. So this is a lie. That variants have different virulence is a lie. Please don't believe it. It is not backed by scientific facts. Point number 2. Variants are infecting youngsters more than adults. There is no age preference for variants, whether it's a youngster or whether it's an elderly, or for that matter, it's a child. There is no difference. So I don't think variants have predilection for younger people or compared to older people. This is the biggest myth, and this is very, very difficult to counter even among doctors. Oh, all these RT-PCR tests, they are missing the variants. That is why you're getting so many false negatives. Remember, RT-PCR that is used across the country now currently amplifies a minimum of 2 genes and most cases 3 genes. And let me also tell you that almost 90% of the tests do not use spike protein gene tests. Even if they use a spike protein gene test, there are other genes where there is no mutation. So the RT-PCR is not missing any variant. If we are getting false negatives, it is because of non-technical issues. A, sample collection. When there is a surge, there can be mistakes. They may not be collected properly. B, it's transported, not stored properly. It is held in room temperature for 2 days before it goes for testing. 3, the large number of tests, there can be mistakes made in the lab, which is human to err. D, the competence of the people who are testing, the quality assurance, the quality control, that is also important. And above all, the most important thing is people who are in home isolation after contact with somebody, they don't get tested, they isolate themselves, they don't monitor, they deteriorate, and they come in the second week with severe symptoms. We all know the best sensitivity for a PCR is between 5 and 7 days after a virus enters the human body. The later after 7 days, sensitivity falls. So you will get a false negative. In serious patients, if nasal and throat swab is negative, the advisory is to collect bronchoalveolar lavage fluid and subject it to PCR. Don't blame virologists and don't blame tests for false negativity. I'll summarize by telling you that genetic variants of SARS-CoV-2 have been emerging and circulating around the world. They will continue to emerge. So that is the, you can take it as a good news or bad news. I leave it to you. Because of its inherent property to make mistakes, as well as large number of people are getting vaccinated, so immune pressure is building on the virus to mutate. How do we detect variants? We need constant surveillance. We need sequencing. Sequencing is a enormously expensive, resource intensive, time consuming technique. It is not a PCR. Please don't expect sequencing labs to give you results today. The turnaround time fixed by INSACOG for sequencing samples is 15 days after it is received in the sequencing lab. Available evidence indicates, this is the silver lining, vaccines protect against variants, albeit to varying degrees. It varies for different vaccines. Some are protecting against all variants, some are protecting against variants, but with less degree of efficiency. As of now, we do not have a vaccine that is useless against variants. The last most important message I want to leave all of you is, variants or no variants, the only way to protect ourselves is to wear masks, maintain distance, and wash hands. Thank God, none of these variants penetrate a mask more efficiently than other variants. First of all, if you wear a mask, you will not get variant or parent virus. So mask continues to be one of the most, and wear it properly, of course, maintaining distance, washing hands. Secondly, even after vaccination, you can get infected, because vaccination gives guarantee of protection against serious disease and death, not against infection. So even after vaccination, you can get infected. I'm currently monitoring at least 6 people in home isolation, my friends and relatives. All of them have received 2 doses of vaccination and completed 2 weeks after that. So this is going to continue for the next 1 year till the entire globe gets immunized, and then also it will continue because variants will keep causing infections. Thank you very much. If you have any questions, I will take them.
Thank you so much, sir. Good evening to one and all, sir. So I'm Dr. Upashri. I express my heartfelt gratitude to our Honorable VC, sir, for initiating this prestigious and very informative program on COVID-19 awareness. And today, especially, I feel even more glad that I have this opportunity to interact with Dr. V. Ravi, sir, such a personality, great personality. Sir, we have this 570 participants who logged in, and we have sir, we have been poured with so many questions actually. So I will take up, I'll read these questions which are there in the chat box. So what I could observe in this speech, many of them have expressed that this talk was very informative, very elaborate, very specific, and very clear to explain the virology part of this COVID. And here I can ask questions. There are more questions based on the vaccine part of it, sir, actually. So first question is, how protective are the vaccination if so many mutations are happening? That is the question asked by many of the participants. And one more question is, which is commonly repeated by many of the participants, is which vaccine is probably best? That is another question which is asked repeatedly. And to what extent we will be protected from these different variants with these vaccines?
Okay, let me reiterate what I said. What is protection? The way we understand protection is, what is the purpose of a vaccine? The purpose of a vaccine is to prevent causation of disease and to prevent mortality due to a disease. Example, polio. Polio vaccine, all of us know. Polio vaccines prevent acute flaccid paralysis and death. But polio vaccination does not prevent infection with polio virus. Same with influenza. Same with any other viral vaccine that you take. Somewhere there is this huge expectation from humanity that COVID vaccine should stop infection. Please get rid of that myth. There is no vaccine in the world today that can prevent infection. Having infection does not mean you are not protected. Protection means you will not develop severe disease and you will not develop death. Let us get those definitions very crystal clear in our mind and then ask the question, how efficacious are vaccines? Yes, when it comes to efficacy, that means ability to cause infection in a person who has received 2 doses and completed 2 to 3 weeks after second dose varies depending on the vaccine. Pfizer Moderna is 95%, but that time mutations were not there when these trials were conducted. Now people are repeating trials in areas where variants are circulating, but that's a very tough exercise. So what they are doing is collecting serum from vaccinated individuals and in the laboratory, like I showed the NIV study, doing neutralization with variant viruses. Now that varies with different vaccines. And I think this is data that is emerging. It is dynamic. Today, I cannot give you this vaccine is effective against this variant. By and large, all papers that have been published in the last few weeks or months have very clearly stated almost all vaccines offer protection against new variants of concern, that is UK, Brazil, and South Africa. But the protection against them offered against infection varies. Some vaccines are very, very low, like AstraZeneca for South African variant, and some of the other vaccines like Pfizer and Moderna not being the same extent effective against the South African and the Brazilian variants. So that will continue to emerge, that kind of data. Covaxin seems promising. It is understandable if you understand the science. Covaxin is a whole virus vaccine. It gives the opportunity for the immune system to recognize and respond to various other proteins other than spike protein. All the other vaccines have totally focused on spike protein, whether it's Pfizer, Moderna, Sputnik, AstraZeneca, Johnson Johnson, Novavax, all of them. And when the spike mutates beyond a particular point, naturally the virus will escape antibody responses to the spike. That way, theoretical advantage Covaxin has more than subunit vaccines.
To ask the question. Dr. K.N. Krishna, please unmute yourself and ask.
Am I audible now?
Yes,
Yes, you are.
Thank you very much, Dr. Ravi, my good old friend. There has been also amongst the doctors lots of literature coming telling that early intervention, vaccination has added to provoking the virus to mutate. What's your opinion on that?
This is a lot of negative opinions building against vaccines. Let me tell you, you need a vaccine because it's a brand new virus to humanity. The only way you get protection to this vaccine is either get infected or get vaccinated. Let me please complete. If you do not get vaccinated, you will get infected. If you get infected, it's a lottery whether you will be asymptomatic. If you're elderly, comorbidities, you have chance of getting severe disease. If you are young and you have no comorbidities, chances are you may be asymptomatic. Okay. And we also know in the last 1 year that people who have asymptomatic infections do not have any clinical problem, but their antibodies wane within 3 to 5 months. That's why you're seeing reinfections. And reinfections will become more common with variants. So you cannot have the cake and eat it too. If you say don't vaccinate, vaccination is causing mutations, the answer is that is not entirely true. To mutate is an inherent property of RNA viruses.
Sir, you are muted, sir. Sir, you are muted.
So
Hello?
please do not blame vaccinations for emergence of
Yes,
mutants.
Gautam.
Do not
Audio is very poor.
blame vaccination emergence of mutants. Even if you did not have vaccination, you will still have variants emerging. That is the inherent property of the virus. Over.
Okay. Sir,
Dr. Rupashri, please, from the chat box.
yes, sir. Yes, sir. Okay. So, which gene has higher sensitivity to detection in human given a choice of so many genes available to test?
There is no single gene that gives 100% sensitivity. That is why multiplexing of genes is carried out in PCR so that your overall sensitivity goes up. If you combine 2-3 genes, your sensitivity will be better. You will not miss mutants, and so your sensitivity will be retained high.
Sir, what is your opinion on checking of anti-SARS-CoV-2 antibodies titer post-vaccination?
I don't think it is required. It is a costly academic exercise. If you have the money and you are curious, please get it done. Clinical trials have shown that whatever is the percentage efficacy, that many percentage of people definitely develop antibodies. In every vaccine that has been developed and used in the last century, there is a small proportion of people who will not respond by antibodies to the vaccine, but they may have developed cell-mediated immunity. So you get tested, it is negative, you start worrying, then you will want a cell-mediated immune test. I'll give you my own example. I had to take hepatitis B immunization thrice before I crossed 10 international units. All 3 times I had to take 0, 1, and 6, a full schedule of hepatitis B vaccine. I never got hepatitis B virus infection, although it ran in my family. I took the vaccine in 1993 when SKF was marketing it globally because of the high risk I had. But I did not seroconvert. Eventually, I seroconvert. So this is a never-ending exercise of getting antibodies done. The best test to get done is neutralization, and that's a very cumbersome test. It's not available commercially. So you will use some standby or surrogate test for it, and that has its own limitations. Over.
So, even if there is a small conformation change in the spike protein, and let's say it escapes the immunoglobulins, but is it possible for the virus to escape the memory cell produced in response to vaccination?
No, you see, it is not so easy to beat the T cell immunity. Mutations in the receptor binding region are very critical for neutralizing activity, but there are a number of peptides from other parts of the virus that stimulate T cell responses. And as I told you, T cell responses measuring is a huge, humongous exercise. If you want to test against all the peptides of the genome that have been shown as T cell stimulating antigens, that is not possible routinely. So yes, as far as antibody, it can escape, but not cell-mediated.
So, Dr. Venkatram, sir is asking, what public health measures could have prevented the spread of this variant in Karnataka?
Very, very difficult public health measures. Difficult in quotes. Wearing masks, washing hands, and maintaining social distance. If these were in place, we won't have variants and we don't have virus mutating. Dr. Krishna asked me about vaccination driving mutation. You don't need vaccination. You allow the virus to surge and 1 million people get infected. Million people every day producing 1 billion particles. You can imagine the number of mutations that will occur irrespective of vaccine. Prevention of infection through behavioral methods is the only solution to prevent mutation of the virus.
The question is from Saida. How true is the spread of word regarding long-term future side effects of vaccine? As per her, which is preventing some of the population to get not vaccinated. Long-term future side effects of vaccine.
GOK, Government of Karnataka, is the abbreviation. God only knows. But I am a person who lives on trust, not on mistrust. I can tell you, I have seen the way safety has been addressed by all vaccine manufacturers. Preclinical toxicity studies have been passed, then only it came for phase 1, phase 2, phase 3. And these vaccines are excepting the mRNA vaccines, all other vaccines have some time or the other. It is not new technology, it is time tested technology. So I don't expect long-term effects with corona vaccines. But post-marketing surveillance or phase 4 needs to be continued. We need to watch out for it. I am not saying that it may not occur. Right now, from the data available, it seems there will not be any long-term effect. Over.
Can we have a question from Galaxy A32? He has asked for a hand raise. Please unmute yourself.
Yeah.
Please go ahead with your question. Okay, Dr. Vatsala Kashi, can you ask?
Hello.
I want to know about the role of anti in clinical practice, not as a public health measure.
What is the question? I didn't hear it, please. Role of what?
Dr. Vatsala, can you repeat?
Role of antibody testing in clinical practice.
Very limited. What the value I see is in sero surveys to know if somebody is exposed to the virus. In terms of clinical practice, the levels of antibodies are not correlated to severity of infection. So there is no correlation. Both mild, moderate, and severe people can either have high titers or low titers. There are many papers published saying that antibody levels do not correlate with disease severity. Antibody detection also is not useful for diagnosis because it takes minimum of 14 days for antibodies to appear. By that time, the person would have infected so many people. That is why RT-PCR is recommended as the gold standard. Over. There are many questions on vaccine.
We are pouring with questions. So,
although this is not the scope of this this talk, what I could do is probably if somebody I can Dr. Mahendra.
Yes, sir. We were about to announce it. Sir.
All the questions.
Yes, sir. We were about to answer this. Sir.
Dr. Rupashree.
Yes, sir.
Ravi sir, with your permission,
Yes, sir.
since most of the questions are pertaining to vaccination, we
Vaccination.
have a scheduled session on vaccination itself. Can we request the participants to come back and raise those questions unless it is such a tearing hurry?
Okay, sir, Dr. N.H. Kulkarni, sir, I have a question. He wants some idea and he wants you to elaborate E-gene value for RT-PCR reporting.
E gene is a gene that is shared between beta coronaviruses. There is common sequences between SARS-CoV-1, MERS, and other milder coronaviruses. That is why E gene was used as a screening test when initial tests were developed. The RDRP and the N genes are more conserved. A combination of E gene with the other genes is helpful rather than using only E gene.
Dr. G.V. Basavaraja, can you ask your question? Please unmute yourself. Dr. G.V. Basavaraja. Gautam?
Madam.
G.V. Basavaraja.
Yes. Sir,
Unmute.
please unmute, sir.
Go for the next person, Dr. Rupashri.
Dr. Vinod Kumar CS, please unmute yourself and ask the question. Dr. Vinod Kumar CS, please unmute yourself.
Sir, namaskar, sir. Ravi, sir, namaskar, sir.
Namaskara, Vinod. Hey.
Sir, I just want to know your opinion regarding CT scan versus RT-PCR. And you clearly highlighted that RT-PCR is more sensitive between 5 to 7 days. But many instances, we see CT score around 20 and RT-PCR negative. And I understand, but there are various factors responsible for RT-PCR result. Just I want to hear from your side, sir.
You are absolutely right. A negative PCR result, there are a number of reasons, and I touched upon them. They are broadly called pre-analytical variables, analytical variables, and of course, post-analytical variables in quality language. Pre-analytical variable, whether specimen collected properly, in the proper medium, stored properly, transported in time, and then tested in time. Those are the sets. Of course, the kind of test people use and the copy test of the person who's doing the test is also important. But natural history of illness in a given patient is one of the biggest variables that affects sensitivity of PCR. And in the second wave, what I have seen, as I mentioned, many people are in home isolation, not monitoring themselves properly. They deterioration occurs between the 5th and 7th day. That's when oxygen saturations fall. That's when consolidation increases. And that's when by that time the virus has already triggered the aggressive immune response. So it disappears. This happened even in H1N1. You will not find the virus in the nose and the throat swab. You may find it in bronchoalveolar lavage fluid. That is why the guidance has been issued nationally and by the technical advisory committee locally, saying that syndromic approach, if RT-PCR is negative, there are a set of pointers that have to be used. One of them is CORAD score, and CORAD score can be used, and the patient is presumptively considered COVID positive and treatment given.
Thank you, sir. Thank you.
The next person, Dr. Rupashri.
Srinivas, can you ask? Mr. Srinivas.
Sir, please unmute, sir.
Yeah, please ask, sir.
Yeah. Hi, Ravi. Srinivas here, dermatologist Srinivas here.
Yes, Srinivas.
Now, after taking this 2 Covishield, can I go ahead and take a Covaxin?
Currently, it is not permitted. I'll tell you why it is not permitted. It's not that we are infringing on your democratic rights. That is not the answer. The answer is when the regulator approved for emergency authorization, he was given data of 2 doses of the same vaccine. Many countries are now doing crossover trials where vaccine A is given followed by vaccine B or vice versa. Those results will come out in 2-3 months. After that, regulators will permit crossover of vaccines.
Ravi, Taramma irudhu. Good. Fantastic.
Thank you.
Dr. G.V. Basuraj. Dr. G.V. Basuraj, please unmute yourself.
Namaskara, sir. Yes, sir. Please go ahead, sir.
Sir, please unmute, sir. Dr. G.V. Basuraj, sir.
Dr. Rupashi, go for the next person.
Okay. Madhuri, please unmute yourself. Madhuri.
Good evening, sir. Sir,
Good
I'm Dr. Madhuri. I just wanted to, can you throw some light on herd immunity? Will
Okay.
it develop or not?
Well, last year we used to all be very, very enthusiastic and gung-ho saying, oh, we will reach herd immunity. Now it looks unrealistic because the way virus variants are emerging, I don't know when the virus is always ahead of us. You may develop immunity to one variant and the next variant you may not. So people now have started talking less about herd immunity, more about vaccine induced immunity.
We have a hand raise from DD6938EB.
That's a new mutant.
Please ask, sir.
So good evening.
Yes, please.
Sir, is there any research being done on effect of alcoholism on vaccination?
No research is being done.
So will it affect in any way?
I'll tell you what I always tell. As a
Yeah.
doctor, I don't prescribe alcohol to anybody.
Okay.
First, point number 2, does alcohol have any extra effect after vaccination adverse event? The answer is there is no evidence. C,
Okay.
should one consume alcohol after vaccination? If so, when? Nobody knows the answer, but common sense if you use, I think it's better to avoid because alcohol, heavy drinkers have problem of nutrition, and nutrition is very key in developing a potent immune response. So
Okay.
it's better to avoid alcohol as far as possible till immunization is over. If not, I guess it's up to the person, but certainly stopping alcohol for a few days is probably good so that the virus will generate the immune response to your body and you don't kill the virus by alcohol. It's alcohol that will kill the virus. This is in a lighter vein. Okay.
Vijay Kumar. Vijay Kumar, can you unmute yourself?
Hello, good evening, sir.
Good evening.
Sir, what are the side effects of this vaccine, sir? Is there any research has been done about this side effects or adverse reactions of this vaccine?
Plenty, sir. Plenty. The internet is full of it. I'm afraid if I start answering the question, you'll have to wait till sunrise. Broadly, every vaccine will produce some side effects. They are classified as minor side effects and major side effects. And some of the major side effects are classified as very severe adverse events, like anaphylaxis, for an example. Common side effects that almost all vaccines will have: pain at the site of injection. Even if you put a needle, you will get that side reaction: swelling, redness, headache, fever, myalgia, and fatigue. The severe side reactions are rash, tingling, breathing difficulty, mainly anaphylaxis manifestations. Some people have gastrointestinal upsets. I've seen that with some of the viral vector vaccines. And some vaccines produce neuroinflammatory syndromes like Guillain-Barré syndrome, transverse myelitis. But they're extremely rare. I think we are slowly shifting to vaccines, Dr. Mahendra, and missing variants. That means virologists and viruses have been spared, or you're still going to attack them?
Okay.
May I request all the participants who have questions with regard to vaccination to please lower your hands. There will be a separate session on vaccination. You can raise your issues there. Kindly restrict yourself to virology and mutants. Thank you so much for your cooperation.
So, Dr. Lalita. Dr. Lalita.
Sir, good evening. It was excellent. You clarified our myths as well. I have one clarification, sir. I just saw the read that children will be affected in the next third wave. Is this genetic variant which is going to cause, or is it the behavior of the children and parents which is going to cause this alarm? Is one question.
Let me answer that question. You seem to be very certain children are going to be affected. Let me tell you, it is a common sense guess. And many of us have made that common sense guess, primarily because the virus will actively seek out susceptible people. If we aggressively immunize everybody above 18, the susceptible pool in the population will be children. And even in the second wave, 10% of infections have been reported in children. And one of the serious complications of COVID, Dr. Basaraj is there. He will tell you from his experience. I think he has seen 4 or 5 already in Indira Gandhi Institute. The multi-system inflammatory disease, which I mentioned, something like a Kawasaki syndrome. Whether a mutant is associated with it, the answer is no. But people are asking the questions, does any particular mutant cause this more frequently? I think that answers will come only in the future, not now. Dr. Basavaraj wants to make a comment. Sir, please unmute. Unmute, sir. Unmute.
Basavaraj.
Unmute, sir.
1 minute, sir. 1 minute. I will do.
Dr. Basuraj, sir, kindly unmute yourself.
Sir, please switch on your video, sir.
He's there. I can
Yes.
say.
No, sir, I don't think he is.
He's there, sir. I can see him. On my screen, he's on your row, Gautam.
Yes, sir. Yes, sir. Sir, please unmute, sir. I have done, sir.
No.
Gautam, can you unmute him for us?
Yes, sir. It's done, sir.
He's now co-host.
I'm sorry.
Yes.
See, basically, the host was not making me unmute. So, sorry. First of all, let me congratulate Professor Ravi. Always pleasure to listen to Professor Ravi, sir. Just one comment on the thing. As far as the multi-system inflammatory syndrome, yes, this is the thing what we are expecting in the pediatric age group, 2 to 6 weeks following in a viral infection, where the 10% of the population of the pediatric is affected. We need to have an eye alert as a pediatrician. That is one. Next, continuation with this, sir said about, Ravi sir said about regarding antibody test. Yes, in pediatrics, especially with multi-system inflammatory syndrome, that is the time where the antibody is going to play the role. Otherwise, in the clinical practice, no need to do an antibody test. These are the 2 comments. Thank you, sir. Sorry, it was a technical issue. That is why I could not be able to unmute it.
Thank you very much for the valuable input, sir.
Dr. Anuradha K.
Hello, sir. Good evening.
Good evening, madam.
Sir, my question is regarding the diagnostic test, sir. Many clinicians ask for CT value. What is your opinion regarding the CT value reporting at the time of RT-PCR?
The ICMR stand is my stand. There is no need to report CT values. There is no correlation between CT value and disease severity. There are innumerable number of examples and papers also published that a low CT does not mean more severe disease and a high CT does not mean asymptomatic disease. Right now in the second wave, I'm stunned to see some people who after 2 doses of vaccination, completing 14 days, have CTs as low as 10, 12, and I saw one report of 8 also. And they are absolutely asymptomatic. So I think you should patiently explain to your clinicians, look, it does not add any value in terms of judging severity. And I've seen patients who have very low CT within 12 days completely clearing the infection. So it's not even related to longevity of infection. So I don't think we should be, because here we cannot correlate it to disease phenotype. Over.
Thank you very much, sir. Very good, clear, informative talk. Thanks a lot.
Dr. Veena Hitchen.
Sir, good evening, sir. After taking the vaccination, can we donate our blood? First dose is over. Second dose is not yet over. Can we donate our blood?
Answer is no, madam. The
No.
blood donation guidelines say that in the preceding 3 months, you should not have taken a vaccine, not because the recipient is going to have problems, because most of the vaccines that we take are killed vaccines. If it is live attenuated vaccine, definitely you should not give, because if there is a very wild, unusual chance of viremia due to attenuated virus that can get transferred. And that is not the issue. The issue is you should get the full benefit of vaccination. And if you deplete your blood by 300 ml, it may reduce your response to vaccination. That is why it is said, do not donate blood after vaccination. I think for COVID, there is some debate going on whether we should lower it to 60 days. I don't think a decision has been taken. Over.
Okay, thank you, sir.
Sir, we have 4 more options. Shall we take it up, sir?
Absolutely, madam. I am at your service. Rajiv Gandhi University service.
Jayashree Bhatt, please ask your question.
Hello, good evening, sir. I've been listening to this conversation and you're bringing up very important points. Thank you very much for that. I've been hearing about this new Andhra strain. I don't know if it has been already discussed and what is the proper information we have about this new very aggressive strain that is being seen coming from some Andhra people.
Okay, thank you.
Any insights?
Thank you very much for asking a question. The lineage that has been reported is a mutation called N440K.
Okay.
And that lineage is in pangolin terminology called B.1.36.29. This lineage has been sensationalized by a non-medical person as an aggressive mutant spreading in Andhra Pradesh. The sequencing was done at CCMB. We also found B.1.3629 in Karnataka circulating between December and February. It has completely died down now. Maharashtra mutant test, double mutant has taken over. This is CCMB, the lab that did the genomic sequencing, has clarified that N440K is not a deadly mutant. We do not have any evidence to link it to disease severity. This is contribution of newspapers, TV channels, and WhatsApp University. Thank you.
Thank you very much, sir. I have another question. So compared to the version, last year's version of the virus, this year it seems to have more predilection for attacking the lungs directly. So in that case, do you think there are several of them who are home quarantined and who are receiving very early steroids? So I just want to debate and ask what is your opinion of
Thank you
use of
very
early
much.
steroids?
Thank you very much for bringing forward a great observation made in WhatsApp University, media, and by lay people. Can you please produce 2 papers that tell me the new mutants are entering the lung more than the nose? This is absolute armchair theory that needs to be rubbished in the nearest dustbin next to you. I'm glad you asked the question. It is not your fault. People are bombarded false information and made to believe. The viruses, all mutants today are going to the lung at the same level and efficiency as the parent Wuhan virus. There is no evidence to say that it is running to the lung, it is escaping the bronchus and the trachea, and tomorrow you will find a WhatsApp information. It went through the skin in the chest directly into the alveoli. Please don't believe such news.
Thank you. Thank you very much for clarifying, sir. Thank you so much.
Dr. Navina J. Can you please ask your question? Navina J.
So good evening, sir.
Hi, Navina, tell me.
It's a wonderful lecture from beginning. I'm here. So I have a question. Yesterday there was a news in the paper. Animals are infected in a zoo at Hyderabad. Should the pet lovers be cautious if asymptomatic people are in the house?
Let me tell you, it is not. Animals getting infected from human beings is news that is 1 year old. The first reports came from Europe. I think it was Denmark. Mink are animals that have very, very thick fur, and you must have heard of mink coats, most expensive coats in the world. Mink got infected from human beings in a mink farm in Denmark.
Yes.
And one of the mutations that was at that time implicated was D614.
Yeah.
And there is a record of minks also infecting handlers. But that is a very rare occurrence because the person who was handling minks was handling a large number of cages and was exposed to probably very high dose of aerosols. Lions got infected.
Yes, sir. Yesterday's news.
This is news. I am yet to see a publication, but I would like to give some credence to news and WhatsApp till credible evidence comes.
Yes, sir. Thank you, sir.
I am not surprised that animals get infected. Whether from animals again it will come back to human beings, I am least bothered because human beings are giving it much more to human beings than animals giving to human beings. You just step out and go to KR market, you will get dose of virus that you will get from 2000 lions. So I think that is the least of our problems. We should be wearing masks to prevent animals getting infected and also getting infection from animals.
Manjula Bhanuri, can you ask your question?
Yeah, sir, wonderful information, sir. Good evening. So my question is like the drugs used for COVID treatment, like remdesivir and all, would they have some impact on diabetics? So just with the drug interactions, so because diabetics and then cardiovascular issues related patients are more susceptible and more facing adverse effects of the virus. So the drugs taken by the COVID patients will have any interaction with these patients, or the diabetics, what they are taking, those drugs will interact with these drugs. Are there any studies regarding that?
I'm not competent to answer this question, but I have read some literature. I can tell you that remdesivir should not be given in people who have abnormal liver function tests and kidney function tests because
Okay.
it has an amount of hepatotoxicity and renal toxicity.
Okay.
I think you should ask it in the clinical section.
Thank you, sir. Once again, thank you very much.
Thank you.
Dr. Preeti Shankar, please ask your question. Dr. Preeti Shankar.
Hello.
Please
Okay.
go ahead.
Yeah. Thank you very much, sir, for the very informative session. I'm sorry, but it is little clinical. Considering that other question which you told that it doesn't directly go to the lungs, so that means that there is no sense in starting steroids early in the treatment.
Absolutely, and that is very clear. That has been
Okay.
clarified by many experts within our state, excellent, competent people, as well as national experts. I think Dr. Guleria went on national channels and said, do not start steroids in the first week. If at home steroids have to be started, the recommendation is to start it after the 5th day based on certain evidences. Dr. Basavaraj wants to chip in. Please allow him.
So what about anticoagulants, sir?
Dr. Basavaraj will answer that question for you.
Okay,
Yeah,
thank you, sir.
just
Thank you.
Yeah, Dr. Manjula, Dr. Preeti, both have raised a clinical question. As far as the diabetes, exaggeration and all, steroid is definitely, it is likely to increase the blood sugar. As far as the remdesivir is concerned, if the liver functions is SGOT, SGPT, anything more than 5 times, you should not use it. That is one. If the GFR anything less than 30 ml per this one, that is the time where you should not use it. And steroid, Dr. Preeti is right in another thing, especially during the viral replication phase, that is in the first 7 days of the illness, the role of steroid does not exist unless there are two situations where you can use an steroid. One, suppose if the patient is having an rise in the acute phase reacting, if the disease process is already in the 2nd week, that is on a 6th or 7th day, that is the time where you can consider a steroid as one of the thing along with an acute phase reactant. And most important is risk stratification. Risk stratification means it is the age, anything more than 60 years, sex, it is a male child, comorbidities like a diabetes, hypertension, chronic liver disease, kidney disease, all those things you need to look at the risk stratification. That is the that's how you can look the steroid as anything. Coming back to the next question is regarding anticoagulant. So that is also once again it has to be looked into the clinical condition. Then you should take a call on that.
Thank you, sir. Madhuri.
Hi, sir. Again, me. Sir, on a lighter note, when do you think this pandemic will end? Lighter note.
When my grandchildren graduate, I don't have a single grandchild now. I'm giving you a lighter note answer.
Thank you, sir. I know.
Dr. Netra. Dr. Netra.
Yeah. Good evening, sir.
Good evening.
It's really wonderful talk you gave and such a big knowledge. As a healthcare personnel, I would want to ask you, there was a article from BMJ as well as Dr. Kang who mentioned that with time we may have to wear masks at home, especially for a healthcare professional when they're going back, when you have children and elderly at your house. What is your take on that, sir?
I would absolutely second it. Given the risk that you mentioned, especially at times when there is extensive transmission. I don't think anybody imagined that Karnataka will have 50,000 cases a day. Bangalore will have close to half of it, 20,000. With this degree of transmission, people who are exposed to the virus continuously, they go home, they are putting others to risk. So maybe the time has come to take a call. I'm not saying everybody at home should wear a mask. It's you have to weigh the risk. Weigh the risk. And the example you gave is very true. I would say yes to wear a mask, or at least maintain social distance if you don't wear a mask. And indoors, virus remains in the air for long. So I've gone on record in many national channels saying that go back to the age-old grandmother practice of opening windows in the morning and putting the fans full blast to ventilate the house. Apartments are also becoming risky because of chutes and aerosols created in toilets hanging around in these tubes. I mean, I don't want to go into that because this is not the forum, but maybe we can discuss that separately, airborne transmission separately.
EP164, please ask your question.
Good afternoon, sir. This is Dr. Subhash here. Mine is a very simple doubt I have. What is the role of heat in killing these viruses? Because I hear a lot of suggestions about taking steam inhalations and some suggestions saying that it will damage the mucosal lining. So what is your...
There is absolutely no evidence that steam kills the virus, number one. There are also other pundits in WhatsApp. I saw a very amusing, hilarious video saying, don't do steam inhalation. It will carry the virus down with the steam. It will take it to the deepest parts of lungs and give you severe disease. Very interesting. I don't know who sits and manufactures these kind of information. Steam inhalation makes you feel good. It opens your passages. It liquefies thick secretions. Enjoy that. Clears pores in your face, sweat pores. You look more beautiful. Enjoy that. But don't imagine steam kills virus. Over.
Thank you.
Dr. Lalita K.
Sir, one more thing. I just wanted to know the receptor binding is not well developed in children. So till what age are they really protected?
This is again studies using very limited number of participants. The argument is that the density of ACE2 receptors reaches its full complement somewhere around after 10 years. Okay. But I must tell you, these are some research studies, and so I do not know. Even if you assume it is correct, they do have receptors. It's not that they do not have receptors. And one thing I must tell you about receptors, I want to remind all of you, the highest density of receptors in the human body is found in the human gut. Almost every cell lining the gut has thousands and thousands of ACE2 receptors. That is why diarrhea is a manifestation in many people you are seeing. So there is evidence to say that the complement of receptors in children is much less compared to adults. But if you look at the second wave, 10% of the second wave, our country data shows it is children. And I think it is children less than 12 years. I'm not talking of the adolescents between 12 and 18. Of course, that isn't going to be a huge group for the third wave because they are mobile and arguably your point that they have full complement of receptors is there. So these are challenges in front of us. Over.
Red MI, please ask your question.
Good evening, sir. Is the new strain anywhere related to the increase in oxygen demand in...
Please identify yourself because your login name is Redmi, which I'm sure is not your name.
So my name is Arpit, sir.
Yes. And your question is whether variants are related to increased oxygen demand.
Yes, sir. In Karnataka, as of currently, more ICU admissions and
no,
more saturation drop.
Absolutely no evidence. It's a very wild armchair theory. Let me tell you, the demand for oxygen. It's not that people infected with mutants are more air hungry than people infected with parent virus. The pattern of the disease has remained the same, whether it's mutant virus or whether it is parent virus. 80% very mild or asymptomatic, 10 to 15% moderate, oxygen goes down, and then 5% very severe, and in that 2 to 3% very, very severe. So 15% will require oxygen. But the second wave is so huge, we don't have so many oxygenated beds, A. And B, the surge occurred in such a short time, oxygen supplies were not there. Imagine some hospitals, all beds have become COVID beds. And almost every bed in a hospital, if it requires oxygen, the requirement of oxygen in that hospital is going to be 5 times, because 1/5 of the beds are oxygenated, largely, roughly, crudely speaking. So it is not to do with variant. It is not that people with mutant are air hungry and the demand for oxygen is more. No.
Dr. Kala Yadav. Dr. Kala Yadav, please ask your question.
Yes, ma'am. Good evening, sir.
Evening. Kala, tell me.
Yes, sir. Sir, in the second wave, we see that a lot of children are becoming positive, including the babies who are born to the newborn, to the positive mothers. Now, my question is, how protected will these children be for the third wave? And do we have an option for vaccination?
The answer is available information in adults tells us that people who get infected develop antibodies. 80% develop antibodies. A small proportion do not develop antibodies. And these antibodies last between 3 to 5 months and they may. Okay?
Yes, sir.
They will get reinfected. If you go by the antibody protection theory, yes, they have a possibility of getting reinfected. Second question, is there vaccines? Currently, trials are going on for children in the age group 9 to 18. Results will be out probably this month end or early next month, and hopefully it will be released for that particular age group. And after that, they will go to the 2 to 9 age group. So right now, vaccination is not an option for children. It's not there on the horizon in India.
So we'll take up last 2 questions. Other questions can be posted in the chat box. We will try to get the answer, and we'll post back in our website as well as in our YouTube channel, and you can refer. And last but one question, NIV BU. Please introduce yourself.
Hello, sir. Good evening, sir.
Hi, Ashok. How are you?
Fine, sir. Fine, sir.
Long time I have not spoken to you.
Yes, sir. Yes, sir. Yes, sir. Sir, it's a very informative and updated talk, sir. And what I could see is we have seen most of the things very scatterly, but you could get everything in a single, and you could give the most updated and excellent talk, sir. Thanks for that, sir.
Thank you.
Sir, just I was thinking that, and we could see since the time the virus initiated, and we could see so fast it is changing because of the transmission mode and all, the lineages and subvariants and all coming up. And we know that S1 and RBD is a site where it is repeatedly changing until that majority of the mutations are at that point. Is there any modeling can be designed so that future it can be predicted, like what could be the next? Of course, one we cannot take directly. There may be series possibilities that can be in somehow helpful more into informatics area. Just a thought, sir.
Absolutely right. There are already 2 groups in India which are doing modeling. They are using the data generated by INSACOG. One is the IGIB group in Delhi, and the other ECMB group in Hyderabad. I know these 2. Probably there are others as well. And this GIS AID database is an open source database. Anybody in the world can access the information. And I'm sure as we are talking, there are hundreds of people who are generating models. There are a couple of papers also. Since I don't understand them, I have not read them, but I can pass them on to you. Yes, there are people who have developed 3D computational models on how the virus is changing, and they also are looking at using it as a predictive tool.
Thank you, sir. Thank you, sir.
06521, please ask your question. Please introduce yourself.
Yeah, good evening, sir. It was a very informative talk. Are you able to hear me?
Yes, please.
Please go ahead.
So this is Dr. Shashikala from Institute of Nephrology. Sir, I have two questions. One is on non-responders in COVID vaccine. Any data on non-responders in COVID vaccines?
I'm afraid no. There are very, very few studies that have looking at immune response in vaccines. My former colleague, Dr. Anita Desai, has initiated a very nice study in Nimhan staff. She is looking at the immune response, pre-vaccination, after first dose, after second dose, comprehensively, both humoral and cell-mediated immunity. I think answers will come from her. But in the vaccine trial phase 3 results that are published, there is data available on what is the seroconversion rates for various vaccines. I can't remember for each of the vaccines how much it is.
Thank you, sir. One more question. What's the effect on immune response post-vaccination in COVID recovered?
Can you rephrase your question? I'm not able to answer.
What is the effect on immune response, post-vaccination immune response among those who have recovered from COVID?
You mean if
natural infection, following natural, yeah.
If I had natural infection and then I got vaccinated?
Yes, sir. Yes, sir.
Okay, this has been answered in the trial results. Almost all vaccines, at least 3 vaccines, I've looked at the trial results very carefully. The vaccine has acted as a booster and their titers are higher.
Thank you so much, sir.
Thank you so much, sir. So with this, we come to the end of this question answer session. We have a lot of questions in the chat box. We will send you the reply if, as soon as possible. And many of you have asked for the speech. So again, I want to just remind you, RGHS is hosting all these talks in its official YouTube, Facebook, Twitter account, and also it is available on the RGHS website. For all the details, you can log in and please check in. And thank you so very much, sir, for your elaborate talk and answering all. And over to you, Dr. Mahendra, sir.
Dr. Dayanand, please okay.
conclude by your vote of thanks.
Yes, yes. Thank you, Dr. Rupashri and Mahendra. Very good afternoon to all of you. On behalf of the Rajiv Gandhi University of Health Sciences, it gives me pleasure to deliver the vote of thanks. We thank our Honorable Vice Chancellor and Registrar for giving us the opportunity to host this training program. So on behalf of the training committee of RGHS, we thank our Vice Chancellor and Registrar. And we thank Dr. M.K. Sudarshan sir for joining us for today's program. In spite of his busy schedule, he has joined. So we thank you very much, sir. And we thank Dr. Ravi sir, who was the speaker for today, for his very clear, informative and excellent talk, and very well taken by our participants. And also you have answered all the questions, sir, from your end. So it was very good session indeed, sir. And also we thank our Dr. G.V. Basuraj, from the professor from Indira Gandhi Child Health Institute, for his remarks during the clinical questions. We thank you very much, sir.
Thank you, thank you, sir. Thank you
very much, sir. Yes, sir. Thank you, sir. Sir, and we thank the Gautam from ECHO India for his providing the very good platform for such a training program. And we thank all the officers of RGHS, Mahendra, Rupashri, for their very good cooperation. And we thank all the participants who have participated till the end. It was a very good 2-hour session, sir. And we thank all of you. Thank you, one and all. Thank you.
Thank you very much.
Thank you so much. Thank you.
Thank you. Thank you.
Sir, very good evening. Good CME for me also. Thank you very much. Thank you. Thank you. I am there right from the beginning till you leave. Thank you. Thank you.
Thank you.
Thank you.
Thank you so much,
Thank you,
Ravi, sir.
everybody. Thank you, Dr.
Thank you.
Mahendra, Dr. Dayanand, Dr.
Thank you,
Rupashri,
sir.
and everybody. Yeah.
Thank you, sir. Thank you, sir.
Thank you.
Gautam, you can close the meeting,
Yeah,
please.
Gautam,
Thank you, Kirti.
Thank you.
Kirti, thank you very much.
Tomorrow's program shall be announced, sir.
Yeah, please do that.
Gautam, please host the flyer. So tomorrow we have another session from Dr. Sattur. Please host the flyer. Gautam? Yeah.
Yeah, it is there.
Yeah. Topic is a critical appraisal of anticoagulation in COVID management. Please do log in at 3 o'clock tomorrow. Thank you. We can close the session, sir, now.
Please. Gautam, you can.